Welcome back to Honors Biology. So today we're gonna just cover again DNA's the discovery of it and the structure. Even though we've already talked about the discovery, we talked about the structure. Just wanted to make this video so you had so you have it in a more notes format, outlined it. This is what you need to know for the DNA structure and the discovery of it. So with that, let's get right into it. So Watson and Crick are credited with discovering the structure of DNA, right? So two important notes there. One, with the structure, they did not discover DNA. They're credited with the discovery of the structure of DNA. They didn't find DNA. They just found the structure of it. And of course, we know there were other scientists at work who contributed to this, Chargaff being one, Pauling being another one, and of course, Franklin. Franklin, without that photo 51, Watson and Crick would have never found or discovered the structure. They would never have pieced it together without that photo. So, but they did win the Nobel Prize in 1962 for their work on the structure. So there you go. So the structure itself, it's a double helix, looks like a twisted ladder. Um, you know, the full name of DNA is deoxyribonucleic acid. So we know that DNA is a nucleic acid. So that's where the Na part of DNA comes from, is nucleic acid. And then the three parts of the DNA nucleotide is the nitrogen bases, and there's four of them, and we'll talk about those briefly. And then the phosphate group that links it, and then the sugar. And the sugar for DNA is deoxyribose. So that's where the full name of DNA comes from, deoxyribonucleic acid. So yes, because it's named after the sugar that's used in DNA, deoxyribose. So that's on the structure. So let's talk a little bit about the nitrogen bases. So there's four bases, right? Adenine, cytosine, guanine, thymine, or thymine. We talked each about the four of them, and they're certainly pairs. They pair together. But how the structurally, how it works is the sugar and the phosphate form the backbone, and we'll talk briefly about that and how that works. And then in the middle, there's the nitrogen bases pair together. And they pair in certain ways. Adenine will only pair with thymine, and guanine will only pair with cytosine. A little bit more about the bases. This is something we haven't talked about yet, and that's the purines and the pyrimidines. So adenine and guanine are considered to be purines. What that means is that the nitrogen bases have two rings to them. So if you look here, here's adenine and here's guanine. They both have two bases, or two rings for their nitrogen base. So we call them purines. Cytosine and thymine, on the other hand, only have one ring. They're single ringed. So we call those pyrimidines. So adenine and guanine are purines because they have two rings, they're double ringed, and then cytosine and thymine are called pyrimidines because they have one ring. So that also influences how they pair together because they're built on base pairing rules. A will pair with T, T will pair with A, and then C will pair with G, and G will pair with C. Now, based on what we just talked about with the purines versus the pyrimidines, adenine's a purine, thymine's a pyrimidine, and guanine's a purine, and cytosine is a pyrimidine. So what that means is that a purine will pair with a pyrimidine, but only certain ones. Cytosine will only pair with the guanine, adenine will only pair with thymine. So a purine will pair with a pyrimidine. That's how that works out. And then Chargaff's rule just states, remember, that since A always pairs with T and T always pairs with A, adenine and thymine will be found in equal amounts in DNA. And since C always pairs with G and G always pairs with C, guanine and cytosine will always be found in equal amounts in DNA. Those ratios are like how much will differ based on what living thing you're talking about. Humans to yeast to bears, each one has different amounts in their DNA, but they'll always be found in equal amounts. The last part of this is I just want to talk briefly about the sugar phosphate backbone. Now, the sugar phosphate backbone, since DNA is made up of two strands of nucleotides, right? It's made up of two strands of nucleotide. So here's one strand of nucleotides, and then here's the other strand. Because of that, they're complementary to each other, right? They're not the same strands. They're complementary to each other. Because of that, we call it. Because of that, right? They run opposite of each other. So when we talk about the sugar phosphate backbone, we talk about them running in opposite directions. More precisely, one strand will run from what we call 3' to 5', and the other side will run 5' to 3'. We get these numbers because of the sugar, which is deoxyribose, from counting the carbons on the sugar. And because of how we count here, 1, 2, 3, 4, 5, 5' to 3' just means that up here is the 5' carbon, and then the phosphate is linked at the 3' carbon. So we got 5' carbon up here, and the 3' carbon, then the phosphate, then the 5' carbon, and the 3' carbon, 5' carbon, 3' carbon. The 3' to 5' strand is the opposite way, right? The phosphate's first, which is then linked to the 3' carbon, and then we got the 5' carbon next, and then the 3' carbon, 5' carbon. So they run opposite of each other, right? One strand's 3' to 5', the other strand's 5' to 3'. This matters when we talk about DNA replication. How does DNA copy itself in the next video? If you have questions, let me know, and I'll see you all next time in class. Bye.